So in today's notes, we're going to talk about pregnancy and a little bit about development in here. So the notes that I have provided for you are going to have more than just the pregnancy and fertilization periods in here. They're a more complete set of notes. However, you only need to worry about the sections that I've put highlighter at the top of those sections. I've opted to just give you the more complete set of notes to have for reference since I already had them made for that, and just update what you needed with the highlighter for this class. So the embryonic period begins with fertilization in the first week of development. You're going to have the genetic material from the haploid sperm and egg that are going to merge together to form a single diploid zygote. So the egg will be in the secondary oocyte form at this point, and this is going to occur in the fallopian tubes. So although there is fertilization in the fallopian tubes, it should move to the uterus to implant. We refer to it as an ectopic pregnancy when fertilization implants outside of the main uterus cavity, and the most common place that it ends up doing it is in the fallopian tube. In order for the sperm to be able to do this, they have to undergo capacitation, where there's some functional changes that are going to prepare its plasma membrane to fuse with the oocyte. So part of what happens is we have an acrosome reaction where we have exocytosis of the acrosome. It's going to fuse the acrosomal membrane with the sperm's plasma membrane so that the contents are outside of the sperm and will expose the inner acrosomal membrane proteins to the sperm's outer plasma membrane. So you're going to have enzymes from the acrosome along with strong movements that help with the penetration. One of the enzymes it uses is hyaluronidase to penetrate the corona radiata with the granulosa cells, and the zona pellucida that is a clear glycoprotein layer between the corona radiata and the oocyte's plasma membrane. So here we can see the sperm cell penetrating the secondary oocyte, where you've got this corona radiata on the outside, the zona pellucida, the plasma membrane of the secondary oocyte, and then the cytoplasm of the secondary oocyte. And then over here you've got the first polar body. So the cortical reaction is a calcium-dependent exocytotic process. You're going to have the contents of secretory granules get released into the perivitelline space immediately after fertilization. This is important because it will prevent multiple sperm from fertilizing the same egg. It's going to have the oocyte membrane depolarized so that another sperm can't get in. And then it will harden the zona pellucida around it. So here is an 1100 times scanning electron microscope picture here showing the head of the sperm coming into the secondary oocyte. And then this is a 250 time magnification of a male and female pronuclei that's under a light microscope. So the oocyte must complete meiosis II. It only does this after fertilization. So it divides into the ovum and a polar body. So that's what we saw up on that first picture, was the polar body that would disintegrate. So you'll have your male and female pronuclei form a single diploid cell. So that diploid cell we refer to as 2N. It's going to have 46 chromosomes. Amphimixis is the fusion of the two different gametes to form the zygote. So when we have dizygotic fraternal twins, they're produced by the release of two secondary oocytes and fertilization by separate sperm. So they are genetically as dissimilar as any other siblings. They just happen to share the uterus. So this here is just showing you have your oogonium, your primary oocyte, and then your secondary oocyte here. And here is that polar body that would be released. So with monozygotic twins, you have a single fertilized ovum. They're going to have the exact same DNA, and there is a separation that occurs after fertilization. So if it ends up being too late of a separation, you can have conjoined twins. So implantation, this is about 6 days after fertilization, and it's going to attach to the endometrium. So this is the uterine lining here, and you can see where you've got this attaching to the endometrium. So it's going to orient the inner cell mass towards the endometrium, and then 7 days after fertilization, it attaches more firmly, and it's going to start to burrow into the endometrium. It's going to do this because the endometrium will become more vascularized and the glands in there will enlarge. So the decidua is the modified portion of the endometrium after implantation. So the regions then are named relative to the site of implantation. So this picture here is just showing this process here. From the first week, you have the ovary ovulating. It will make its way into the uterus. Somewhere along the way here, you've got fertilization that will occur. You do have a little bit of cleavage that happens there, becoming multiple cells, and then implantation here after about 6 days. And this is just a little bit larger picture of that process happening. So placentation, this is the formation of the placenta. This is going to begin about at 12 weeks, and there's two parts. The fetal portion is formed by the chorionic villi of the chorion, and then the maternal portion is formed by the decidua basalis of the endometrium. So this is going to allow oxygen and nutrients to be able to diffuse from the mother to the fetal blood, while carbon dioxide and waste is going to diffuse from the fetal blood to the mother blood. So it will put nutrients and oxygen into the baby. It's going to take the baby's waste and put it back into the mother's blood supply. Fetal hemoglobin is going to bind oxygen more strongly than maternal hemoglobin, which is what allows this to happen. So this is not a protective barrier. It does allow some microorganisms, drugs, and alcohol are able to pass through it. So there's a connection between the embryo itself and the placenta through the umbilical cord. You have two umbilical arteries that are going to carry deoxygenated fetal blood to the placenta, and then one umbilical vein that's going to carry oxygenated blood away from the placenta. So after birth, the placenta will detach from the uterus, and the placenta is delivered afterwards. So this is showing here your umbilical cord with your umbilical veins, the mucous connective tissue, and the amnion. Here's your chorionic villi portion, the decidua basalis portion. And then this is showing here with the fetus inside. And then this is a picture of a placenta here. So this is one that has been preserved. Typically a placenta is very red after birth. So we have some hormone changes that are going to go on in here. So the human chorionic somatotropin, or human placental lactogen, is going to be produced by the chorion. That will help to prepare the mammary glands for lactation. It also will regulate certain aspects of the fetal and maternal metabolism. We're going to have corticotropin releasing hormone produced by the placenta. So in a person who is not pregnant, you do get corticotropin releasing hormone produced by the hypothalamus and then secreted out of the anterior pituitary. One of the thoughts is this is part of a clock establishing the timing of birth. It will increase the secretion of cortisol that's needed to mature the fetal lungs and then also produce surfactant. So the main hormone you'll hear about is human chorionic gonadotropin. It's a hormone that's produced by the placenta during pregnancy. This is found in the urine or blood around 10 to 11 days after conception. It helps to support the pregnancy by allowing for production of progesterone, which will prepare the lining of the uterus for implantation. That is also the hormone that is measured in a pregnancy test. So this just compares the different hormones that would be coming out of the placenta. You've got the HCG that's going to be released early, helping to prepare the placenta. Relaxin, this is going to help with increasing the flexibility of the pubic symphysis, the pelvis being able to change shape and dilate the cervix during labor. The human chorionic somatotropin, preparing the mammary glands for lactation, increasing protein synthesis, and then the corticotropin releasing hormone helping to establish birth timing and the secretion of cortisol. And this lets you see the levels of the hormones. So the HCG is early on, progesterone and estrogens increasing later on in the pregnancy. So labor or parturition is the process of expelling the fetus from the uterus through the vagina. The onset is determined by interactions between several placental and fetal hormones. We know there's at least 5 factors that seem to affect the process of labor and birth. Sometimes they refer to them as the 5 Ps, looking at the passenger, which is the fetus and the placenta, the passageway, things in the birth canal, the powers, the contractions, the position of the mother, and a psychological response. So you have to have the levels of estrogen rise to overcome the inhibiting effects of progesterone on uterine contractions. So the high level of estrogens will increase the number of receptors for oxytocin on the uterine muscle fibers. The oxytocin is going to be the one that stimulates contractions. Relaxin will increase the flexibility of the pubic symphysis and dilate the cervix. And labor is one of the few positive feedback cycles we have in the body. What's going to happen is, as the contraction force of the fetal head goes into the cervix and stretches, the stimulated stretch receptors are going to cause the release of more oxytocin, which is going to be more contractions for more stretching. So it feeds into itself, and it will keep intensifying the contractions until the baby is actually delivered. So the cycle is broken when the stretching stops as the baby exits. True labor begins when the uterine contractions are at regular intervals. And then as the intervals shorten, the contractions are going to intensify. So there's 3 stages. You have the birth of the baby and the delivery of the placenta. So you've got active labor, birth of the baby, and delivery of the placenta. So some things with the placenta. Placenta previa is when the placenta covers the opening of the mother's cervix. Placenta abruptio is when the placenta separates from the inner wall of the uterus before birth. So with a breech presentation, normally when the baby comes out, it's supposed to come out head first. This is when the baby's buttocks or its feet or both are in place to come out first during birth. So something other than the head. So in the past, occasionally breech babies would be delivered vaginally. nowadays it's very, very unusual to have them be delivered vaginally. They're usually going to be delivered via a C-section or cesarean section, which is a surgical procedure used to deliver the baby. An incision is made in the abdomen and the uterus. So the common indications for doing a cesarean include labor dystocia, so abnormally slow labor, abnormally indeterminate, or what they used to call non-reassuring fetal heart rate tracing, fetal malpresentation, so it's not in the right position, multiple gestation, so if there is multiple births, or suspected fetal macrosomia, which is a large baby over 8 pounds 13 ounces. So they do occasionally deliver larger babies. So risks of C-section, it's a surgical procedure, so there's always risk of infection, postpartum hemorrhage, there's risks with anesthesia, blood clots, other surgical injuries, and there's increased risk during future pregnancies. So generally, if you have a C-section for one, you're going to end up having a C-section for all subsequent babies. So for lactation, this is the secretion and ejection of milk from the mammary glands. Prolactin is going to be the principal hormone promoting the synthesis and secretion. It's going to come from the anterior pituitary. The levels of prolactin are going to rise during pregnancy, but progesterone inhibits the effects of prolactin during pregnancy. After delivery, the inhibition is removed because the estrogen and progesterone levels fall. So then the principal stimulus maintaining the prolactin is the suckling action of the infant. So actually breastfeeding. If that doesn't happen, then it won't maintain the stimulation of the prolactin. So impulses from the stretch receptors decrease the release of prolactin inhibiting hormone and increase the release of prolactin releasing hormone from the hypothalamus. So you have a negative feedback loop here. The suckling response of the baby with touch sensations in there, going to go to the control center in the hypothalamus and posterior pituitary, increasing the oxytocin. Then you have contraction of the mammary glands to eject the milk. And then positive feedback in here. The milk availability encourages continued suckling and continued milk production. So when the baby stops suckling, it will work to break the positive feedback cycle. So oxytocin is going to cause the milk ejection. Suckling, hearing the baby cry, touching the mother's genitals can initiate this, so it also is the one that did the uterine contractions in there. There are other effects of oxytocin fostering bonding between the mother and baby as well. So colostrum is going to appear before the true milk on the 4th day. Colostrum is really important for the baby because it's going to have some important antibodies in there, so it's very healthy for the baby. Lactation is often going to block the ovarian cycle for a few months after delivery. So a person may not have a menstrual cycle. While it may happen, it is not guaranteed that it would happen, and it's certainly not something that a person would want to rely on as a means of birth control. So a lot of people have siblings that are 10 months difference between them because that plan of using it as birth control doesn't work very well. The primary benefit of breastfeeding is nutritional, but there are other benefits also.